Thank you. Okay, so I'm going to change topic again, and my background is mainly biological, and I will give you a talk that is kind of methodological. So we will see together how imaging and quantitative analysis can be integrated to study neuronal morphology and synaptic organization. And I would like to start from this. So the idea that organization and functioning of neuronal circuits is strongly related to the structure of neurons is actually very old. In the late 19th century, Golgi stainings and Ramon Cajal drawings allowed scientists to visualize that different morphologies of neurons can be related to the brain region to which the neurons belonged. And these intuitions are still valid today. At larger scales, the brain is highly complex, structurally, molecularly, and we have modern technologies that allow us to classify cell types depending on their molecular signature. But despite the power of the omics approaches, actually, neuronal morphology cannot be fully captured by transcriptomic cell classes. So we need to have reproducible and robust ways to quantitatively describe a neuronal structure. And this brings us to the challenge we will see today. Imaging still matters for at least 3 reasons. It complements transcriptomic cell classes, it preserves the spatial context and organization, and it links structure to connectivity and function. However, turning images into reproducible readouts is still challenging because neuronal structure cannot be defined by a single parameter. It is related to branching, extension, complexity, or specific compartment effects. Moreover, measurements depend on multiple factors, such as the image acquisition and quality, the segmentation choices, tracing rules, the cell density of neuronal cells, even in culture, crossing neurites, and also batch-to-batch variability. So we need to find a way to be able to extract measurable, comparable, and also biologically meaningful structural phenotypes. And in this framework, it is important to keep in mind that morphology is not just geometry, and in the brain, neuronal structure is shaped by communication with other cell types. This communication, this interaction can be mediated by direct physical contact or extracellular signals, and they have a strong impact on neuronal development, growth, and synapse structure and function. I'm going not to enter into the molecular details of this mechanism, as this is not the scope of the talk. This is just to let you know what is the biological logic behind the experimental framework I will show in a moment, because in the lab, we work with primary neuronal culture. We work with immature or mature neurons, and we expose this neuronal culture to soluble molecules that are directly added to the neuronal medium to understand how these extracellularly derived factors can impact on the neuronal morphology. Maybe. Sorry. Oh. Okay, on the neuronal morphology, or can shape the synaptic organization. So for the first part, I will focus on neuronal morphology. We will see together how this can be quantified using specific assays. Maybe, I don't know why it's, okay. So when you work with cell in culture, you have multiple cells, and the first thing to do is to find a way to isolate single cells, to be able to analyze them, not to have interference from crossing neurites coming to neighboring cells. And another important thing is to use complementary metrics to describe the morphology of these cells. Generally, people use this analysis, which is called shoal analysis, to obtain information about branching as a function of distance. Indeed, with this analysis, you can obtain the number of intersections at increasing distance from the soma. And then this analysis is usually complemented by other types of analysis. Here you have the fractal analysis, normally run using the fractal plugin, and it gives you an idea of the overall geometric complexity of the object you are analyzing. This is just to show you how results can be quantitatively observed. So we analyzed our two conditions, and we observed that the neurons cultured in enriched media presented higher complexity and higher branching over different distances from the soma. And the same result was also confirmed when we did the fractal analysis. And you can see here how these two different analyses give you different, non-redundant information. On one side, you have an index that recapitulates the overall complexity of the object. On the other side, you are also able to locate where, spatially, where the measured differences are concentrating. At that point, once you have global information about complexity, you may ask, okay, but what is the precise structure of the neurons that is driving the phenotype we are observing? To answer this question, people generally use neurite-based tracing tools, such as NeuronJ. These tools allow you to obtain the dendritic arbor of the cell type, and beside that, you are also able to assign different colors and therefore obtain different measurements for the different types of neurites that compose the neuron based on the trajectory and also on the branch order. In this way, you can move from a global information to a neurite-specific information. I should say also that actually all these tools are semi-automatic, and semi-automatic pipelines generally are time-consuming and also operator-biased. So since we are looking for reproducible, robust, and standardized workflows, I can tell you that it is possible to scale up these workflows using machine learning-based approach. So this is just an example of the same neuron I showed you before traced manually that we were able to reconstruct after proper training of machine learning with a large data set of image. And this is, I think, an important way to consider in order to go to the direction of obtaining standardized and highly accurate measurement of neuronal complexity. So to sum up this first part, we saw together these three different approaches that give us complementary information ranging from branching, complexity, and compartment-specific effects. And the main take-home message here is that no single matrix is fully sufficient on its own to be able to capture morphology. Then, going to the second part of the talk, which is mainly related to synaptic organization, I can tell you that even with this type of analysis, there are some analytical features that are shared with what we just saw. So for example, again, you need to isolate single cell and to have anatomical scaffold, which is the dendritic arbor on which the synaptic measurements can be anchored. And then you can overlay the structure with specific synaptic markers. These are just proteins that localize specifically on the presynaptic compartment or postsynaptic terminal. And when these two compartments are overlapped, we just consider to have a synapse. But then, now, how do we decide which puncta belong to that structure and which combination should be considered as synapses? Again, different tools can answer the same question in different ways. So, for example, we can rely on very specific tools for neurons, such as this SynapCamJ plugin, which quantifies the pre- and postsynaptic puncta over the defined neuronal structure you provide. And as an output, It provides you the length of the dendritic structure, the number of synapses, and the synapse density, keeping into relation these two parameters. But then you can also decide to rely on more general tools, such as the ComDat plugin, which is not really specific for neurons. It is more related to object and puncta detection. It needs to have as an input information related to puncta size and thresholding parameter, and then it gives you the number of puncta detected across different channels. And in our case, we can consider presynaptic, postsynaptic, and spatially colocalizing puncta as number of synapses. And even in this case, we analyzed our different condition using the two independent plugins. And with Synapse Count J, we were able to observe that neurons acutely treated with these extracellularly derived factors showed higher number of synapses. But then when the same data set was analyzed with ComDat, actually we were not able to recapitulate the same result. Indeed, we didn't observe significant difference at first, but then the warning sign was related to this, so the fact that even the positive control was not responding to the treatment with this molecule that is strongly reported in the literature to increase synaptic matching, and this was not able to be recapitulated by our analysis. At that point, we came back to our data, and since this plugin, as I told you, is strongly based on particle size that the operator provides as an input, we were relying on something that was published in the literature, but then maybe this was not really related to the experimental paradigm we were working on. So we went back to our parameters, we adapted segmentation and thresholding choices, and then we checked quantitatively and qualitatively the results comparing control and our positive condition. And after doing that, we were able to recapitulate the same effect even with this other analytical tool. Since I told you before, with this other plugin, it is possible to retrieve information about separate channels, we can also say that our treated neurons are not only showing higher number of synapses, but the effect is mainly driven by the postsynaptic compartment. And yeah, before going into conclusion, I also would like to flag you this other plugin, which is Symbot, that has been very recently published. And this is, I think, very powerful because it goes toward the same direction I told you before for machine learning. So it allows to automatically quantify the number of synapses over the neuronal structure. So going into conclusion, I would like to leave you with this consideration. Complementary and well-calibrated pipelines are essentially to obtain meaningful results. And also parameters do not always generalize. Even if published in the literature, check very well if they are right for your experimental paradigm, and also try to include the proper controls in order to prevent the analytical artifacts. Then automation can improve reproducibility, that is what we are searching for, and also reduce the bias. And finally, these structural-based imaging tools can be integrated with functional platforms, and in this way it is possible to move from structural phenotypes into functionally testable hypotheses. And I would like to show you this video that can give you an idea about the fact that today we are able to interrogate the same neuronal preparation, both structurally and functionally. And using the proper markers, we can correlate the neuronal structure, synaptic organization, and also the network activity. Now, I finished. I would like to thank all the FNIP organizing committee, Nicoletta for this opportunity to have this talk, and my lab mates, and you for the attention.